It's a great pleasure to be here today. I have to say, though, that pleasure is a little bit tempered. I'm going to share with you a little bit of work which my lab has been doing, centering on single gene disorders of insulin action, and I'm going to spend some time also discussing PI3 kinase and its role in metabolism. And so I confess to some sense of the icy hand of fear clenching my bowels when I looked at the plenary schedule and saw that Steve O'Reilly, my erstwhile mentor and perhaps the dominant figure in the study of single gene forms of insulin resistance, is talking tomorrow. Lou Cantley, who discovered PI3 kinase and has been there at most of the key steps forward in its development, is talking first thing tomorrow morning. And to cap it all, Ron Kahn is coming later in the week, and Ron's lab has really done most of the work using murine models to dissect the role of different parts of the PI3 kinase complex in metabolism. So it's very much with a sense of being, of tiptoeing among giants that I offer you a few thoughts today. So I'll kick off with my favorite caricatured view of the world of what I sometimes call common metabolic disease. And by that, really, I mean the things that we see related to obesity in our endocrine practice. And so, although I have included disease down here, which I take to be the more mechanical consequences of obesity, I'm predominantly talking about type 2 diabetes, fatty liver, dyslipidemia, atherosclerosis, and soft tissue overgrowth up to and including cancer. Now, my three-stage scheme towards these diseases, first of all, relies on chronic positive energy balance, which is a neurobehavioral problem which I don't work on. It then critically relies, and this is a very defensible but not uncontested view, this view of adipose tissue as being critical in common disease, but I believe it then depends critically on a very heritable capacity of our adipose tissue to expand safely to store energy. So, in other words, some of us can lay down lots of fat tissue without any metabolic consequences, while others become sick relatively quickly. So in those who do have that mismatch between energy excess and buffering capacity, they proceed to a state which is often called insulin resistance, but as I'll reflect, insulin resistance is a very vague term. Insulin is a very pleiotropic hormone that does many, many different things in cells and tissues, and so to define it simply with respect to one parameter is potentially misleading. And that intermediary metabolic state is what is associated in great detail and in large population studies with the diseases which I'm interested in. So to crystallize from that some questions. First of all, I'm interested in contributing to understanding what the determinants of our innate capacity to expand our adipose tissue are. Why do some of us get sick while others don't? As I've said, I'm interested in the notion of what insulin resistance actually is. Can we subdivide insulin resistance into sub-syndromes? And perhaps closest to my heart is trying to understand whether insulin resistance does indeed cause the diseases with which it is associated, and which is a claim which is often made, or are some of them merely epiphenomena? And the tool that my lab uses is very much the study of patients with rare and extreme single gene disorders. And in the talk today, I'm going to divide what I'm going to tell you into three parts, none of which, I should say, have a very firm conclusion. This is very much more a matter of describing to you the evolving areas of interest which make me open my laptop in the evening and which give me something to think about as I cycle to work. And so the first of these is the power of human genetics to show us unexpected stuff, and that's tremendously powerful. Second, I want to describe to you the use of well-known genetic variation as a form of Mendelian randomization applied to rare single gene defects. And finally, I want perhaps to move a little bit off-piste and suggest to you that even in areas where cellular mechanism has been studied in fairly exhaustive detail for several decades, there are still interesting observations to be made in human genetics. So I'll start where we always do with a patient, and this is a lady who I first met when she was about 15 or 16 years old. She is compared here to a slightly unfair 1 m male control, but nevertheless I think you get the sense that in adult life she is extremely short, and that severe growth impairment was apparent since her time in utero. So she has a form of primordial dwarfism, and allied to that I think you can see some degree of facial dysmorphism. Now, that's not all her syndrome entails. She also had some evidence of mild spontaneous chromosomal damage. That wasn't a clinical problem, but that was picked up several times in her early years of life. Her dominant medical experience, however, throughout her life has been the travails associated with very, very severe insulin resistance. And looking back through the records, some of the clues were there from about the age of 3 or 4 onwards. But it was only around the age of 11 that this was formally investigated and documented. And you can see in the few brief notes I've made here exactly how insulin resistant she was. So at the age of 11, with a fasting glucose of 4.7, her insulin, which should be around 30, 40, 50 picomoles per litre, was 4,000. And over the ensuing years, as her diabetes was treated with insulin, she required up to 25 units per kilo per day. Reflect on what that would mean for you. And when we evaluated her more formally at the age of 17 years, she was not frankly lipodystrophic, but she was unequivocally thin, as you can see from her body fat mass by DEXA scanning and leptin. She also had primary gonadal failure, but she didn't have any clinical evidence of premature aging or malignancy. Now, these disorders are extremely rare, but we had the opportunity to study a further patient who was sent to us by Jean-Claude Carrel in Paris. And you can see she essentially shared the same syndrome. There were some differences. She had congenital blindness, but she had severe primordial dwarfism, extremely severe dyslipidemia and insulin resistance with pancreatitis due to hypertriglyceridemia on at least 2 occasions, as well as primary gonadal failure. She developed heart failure when she was 26 years old and died of a sudden cardiovascular death, which wasn't very well characterized when she was 30. Now, to cut a long story short, we applied exome sequencing to our first patient and her parents and simply looked for either de novo or compound heterozygous mutations. And we only came up with one plausible candidate, and that was in this gene, NSMCE2, where we found 2 different frame shift mutations. And you can see from the family of the first proband that she was the only one in the family who had those 2 mutations. Now, NSMCE2 encodes a sumo ligase. Sumo is a small peptide like ubiquitin, which is stuck to other proteins to modify their function. And this particular sumo ligase sits in the middle of a large protein complex called SMC56, which, to cut an uncertain story short, plays a role in resolving tangled DNA during the process of DNA damage repair. And it looks very much like cohesin and condensin, 2 other nuclear protein complexes, which play important roles in DNA repair. Now, one slightly unusual aspect of this case was both our patients had precisely the same 2 mutations, and that caused quite a good deal of worry in the lab. But we independently secured samples from the parents in Brittany and in North Wales. And in fact, some haplotype analysis suggested that there was a founder effect with a common shared ancestor about 4,000 years ago. Now, I'm not going to go into the cell biology in great detail because it's actually not very well understood. But suffice to say, in primary cells from our patient, we were able to show that there's very little or no NSMC2 protein. This simply shows increasing amounts of protein being loaded. And we went on to demonstrate that there was a highly significantly increased level of micronuclei and nucleoplasmic bridges visible when we looked at the cells. And these are both non-specific indices of excessive DNA damage. So there was some evidence that DNA damage wasn't quite being handled correctly in these cells. And when we used a lentivirus to conditionally express the wild-type protein under the control of doxycycline, you can see here the wild-type protein being turned on in both the patients and the control cells. We were able to abolish that excess. So you see here the excess of micronuclei, in this case, being completely abolished by addition of DOX and induction of the wild-type protein, whereas inducing a ligase-dead protein actually worsened that phenomenon. So that's by no means the final word on what that protein does, but it was quite strong evidence that we did indeed have a functional defect. And working with Ines Barroso at the Sanger Institute, we used morpholinos to knock down the protein and confirm that this produced dwarf fish. And importantly, re-injecting wild-type mRNA rescued that size, whereas re-injecting mRNA from our 2 mutants failed to rescue it. So it's fairly clear, although we only have 2 patients, that we have the correct defect. So that truly is one of those odd genetic observations. What does it mean? Should we be surprised to find ourselves in the territory of DNA damage repair and insulin resistance? Well, perhaps not, because of course 2 syndromes have been around for many years which also feature dyslipidemia and severe insulin resistance, among other features. And those are Werner's syndrome, very enriched in Japan and due to mutation of a DNA helicase, which is one of the more faithful progeria syndromes in the sense that it reproduces many of the features of aging in the 20s. And some years ago, we found a patient in the UK whom we diagnosed with exome sequencing, probably some 10 or 15 years before it would have been diagnosable clinically otherwise. And you just get the beginnings of the sense that between 7 and 16 years, she's beginning to develop the characteristic loss of fat and muscle from the extremities, which generally progresses horribly. Bloom syndrome is another premature aging syndrome with photosensitivity and dwarfism. Again, it's due to mutation of a DNA helicase, and again, it features insulin resistance, dyslipidemia, and diabetes. And the final member of the quartet is more novel, and this was a syndrome which was first described in Texas by Abhimanyu Garg some 5 or 6 years ago now. And it's a syndrome which features fairly dramatic loss of both muscle and fat from the extremities, as you can see here, often progressing ultimately to calcinosis and ulceration, together with some other features, including deafness and male hypogonadism. In Andrew Hattersley's lab, with whom we collaborated, exome sequencing of a further patient with the syndrome was done, and this established the cause to be a single amino acid deletion from POLD1. POLD1 encodes a DNA polymerase, and this amino acid deletion lies in the polymerase domain. And the very same mutation was found in 4 further patients, and in no case was it inherited from the parents. We worked with Larry Loeb in Seattle, who did some lovely in vitro studies of this polymerase mutation. And without dwelling on the detail, you can see that while a wild-type enzyme shows ability to polymerize a small DNA template, this is run on a polyacrylamide gel, the mutant protein was not able to polymerize at all, while it had preserved ability to reduce the size of the template. In other words, it had preserved exonuclease activity. So this story ends here, but we are now in a situation of having an evolving group of disorders which unify defects in DNA damage repair and sometimes DNA replication with severe insulin resistance. And these disorders are otherwise rather heterogeneous, although you can see this further disorder, which I haven't discussed, features DNA polymerase zeta, a different type of DNA polymerase, which features an even more severe degree of dwarfism. There's no dwarfism in the POLD1 syndrome, which I mentioned to you, and it's modest at most in Werner's syndrome. So is this just the scrapings of genetic oddity? Is there any coherence to this? Does it tell us anything? Well, my favorite model is that this is beginning to define for us the genetic architecture of our adipose expandability, and it's probably telling us something about a vulnerability of adipose tissue to DNA damage. And I like to think about this together with the consequences of early childhood cancer treatment, which I think is an under-recognized but important aspect of late effects practice. And this is out there in the literature, but it's scattered in various different places. So in our cohort of severe insulin resistance, we've collected probably 5 or 10 patients with very severe degrees of insulin resistance and very severe dyslipidemia, and each of them had whole body irradiation as part of cancer treatment when they were 3, 4, or 5 years old. And their metabolic derangement doesn't characteristically start until around puberty, at which stage girls naturally lay down fat mass. And it, although is sometimes confounded by growth hormone deficiency, it's quite clear this is independent of growth hormone status and does occur in growth hormone deplete girls. And in fact, for other reasons, this was modeled in mice a few years ago. So this French group, with an interest in the behavior of fat peritransplantation, demonstrated that if you irradiate leptin-deficient mice, highly obese leptin-deficient mice, at first sight it appears to be a good thing because they put on less fat over the ensuing weeks and months. However, they become very metabolically sick. They become more insulin resistant, more dyslipidemic, and unwell. And that's a consequence of impaired proliferation of their fat cells and increased cell death. So I think we are beginning to look at 2 sides of the same coin. I think if you either impose a genotoxic stress from outside or you confer increased sensitivity to endogenous genotoxic stresses, there appears to be an Achilles heel of adipose tissue or its precursors, which constrains its expandability and makes us very prone to metabolic disease. And that's something we're increasingly going to be looking at in mouse models. So the second application of human genetics I want to discuss is Mendelian randomization, which is a rather formidable and complex way of saying something very simple. So as applied to this problem, let's imagine obesity and an obesity-related disease, let's say polycystic ovary syndrome. And let us hypothesize that the X here is insulin resistance. In other words, that obesity drives PCOS because of insulin resistance. Now, the best way to test that, of course, is to induce a state of insulin resistance and see if polycystic ovary syndrome results. But that is either impractical or unethical in most human settings. So instead, you borrow from what nature has provided us. You look for natural variants which produce a primary form of insulin resistance, but which have no independent link to the ovaries or to ovarian function. And you ask the question, are people who we find, because they've got that primary insulin resistance, do they have PCOS? And if they all do, then that's rather strong evidence for it. And if they don't, it's counter-evidence. And I just very briefly introduce you to a few of the sort of key cast characters for this end of the talk. So the obvious form of primary insulin resistance are those patients with insulin receptor mutations, and they've been known about since about 1987. There's more than 150 mutations known, collectively several thousand patients in the world. And my observations are mostly based on those patients who have type A insulin resistance with autosomal dominant diseases, by far the commonest form, who otherwise are relatively normal in terms of growth and shape, as you'll see from this girl along at the end, and quite distinct from the horrible recessive insulin receptoropathies, which leads to early death. Now, we do have one or two other cards in the box, and one of those is the single family with a loss-of-function AKT2 mutation, and I'll talk you through this insulin signaling pathway in more detail anon. This was identified by Steve O'Reilly in our part of the UK around 10 years ago, and we still have available 2 patients from that family to study. So the first observation to make is that although our patients with insulin receptor mutations have the most severe degree of insulin resistance there is, they never have dyslipidemia, they never have elevated triglycerides or reduced HDL cholesterol, and that is true now in 70 or 80 patients who we have analyzed. In contrast, patients with primary defects in their adipose tissue, or in fact, interestingly, in that AKT2 protein, have an exaggerated form of the common type of dyslipidemia. They have severely elevated triglycerides and suppressed HDL cholesterol. And this graph is taken in the fasting state. And that's backed up by studies of liver fat. So these are images taken from magnetic resonance spectroscopy, where you can see the triglyceride proton peak next to the water proton peak. And in our patients with insulin receptoropathy, who remember are exquisitely insulin resistant, there is no more fat in the liver than you see in healthy insulin-sensitive controls. In contrast, patients with the commonest form of lipodystrophy or with AKT2 mutation have livers which are like foie gras. They're full of fat, very, very unhealthy. Now, a low plasma adiponectin level certainly isn't a disease, but adiponectin is now the subject of many thousands of papers. It's a highly abundant protein derived from fat tissue, and it has been suggested to be one of the mediators of insulin resistance. Now, I'm not going to comment very much on what adiponectin does. I have some doubts about that, except to say we find it tremendously useful. So it's low in common forms of insulin resistance. It's low in any form of severe insulin resistance caused by a problem with the adipose tissue. And yet, extraordinarily, in patients with insulin receptor mutations or antibodies against the insulin receptor, it's not just much higher than the other severely insulin-resistant patients, it's actually much higher than healthy controls. And I've had to curtail this graph because this goes up to 100 milligrams per liter, 0.1 grams per liter of adiponectin. So we now use this practically to screen our patients with insulin resistance and to triage them for insulin receptor screening. So those are actually very simple observations, but they tell us that insulin resistance is absolutely not homogeneous. So there are different types of insulin resistance. There is if you like, a proximal insulin signaling type of insulin resistance, where there is no fatty liver or metabolic dyslipidemia, where adiponectin is not low, but where other features are very severe. And for reasons which are complex and which I don't understand and won't therefore go into today, just stepping down the insulin signaling pathway 2 steps actually doesn't do that. It reproduces the whole gamut of common disease. Now, we very much want to look at further genes in the pathway to see if we can understand that discordance further. And fairly recently, we have been given that opportunity with the discovery by 3 different genetic groups of the genetic cause of short syndrome. And this describes short stature, hyperextensibility of joints with hernias, ocular depression, Rieger abnormality, which is a developmental abnormality of the iris, which you can perhaps get some sense of there. And although not part of the acronym, partial lipodystrophy and insulin resistance are also known to be a common feature of this syndrome. Now, the causal gene is PIK3R1, and in all cases studied so far, the mutation lies in the C-terminal domain, and PIK3R1 forms part of the regulatory subunit of, is the regulatory subunit of type 1a PI3 kinase. And again, I'll talk you a bit more through the pathway later on, but this is intermediary between the insulin receptor and AKT2. It was, I confess, a little bit of a blow to us, these 3 papers, because we had found another patient with a truncating mutation 2 or 3 years before that with severe insulin resistance, but we hadn't been able to find those critical 2nd, 3rd, and 4th patients to publish. Even more frustrating, we did so very quickly in short order after those publications, quite independently of the publications. But they allowed us to ask the same question. So what does it look like when you lose PI3 kinase function? Do you look as if you have a proximal insulin signaling defect, or do you look as if you have an AKT2 defect? And the answer is actually clear. So especially our 4 teenage girls here, as you can see, very severely insulin resistant, not unduly diabetic at this stage, with insulin levels which are at least an order of magnitude above normal. They have very high testosterone levels. This is not at all surprising in our cohort of patients with severe insulin resistance. We see levels occasionally as high as 10 or 15 nanomoles per liter in girls. And yet their lipid profiles are entirely normal. And moreover, in a single patient where we were able to study it, we could hardly detect the liver fat by magnetic resonance spectroscopy. And using deuterium incorporation into palmitate, we could hardly detect de novo lipogenesis in the liver. Now, I'll set aside an interesting caveat around the first patient who actually has an independent mutation in the adiponectin gene, and point you towards the other patients who have levels of adiponectin which are exactly in keeping with our insulin receptor patients. So this is where our Mendelian randomization stands. So either PI3 kinase or insulin receptor patients produce this different type of insulin resistance, which is uncoupled from some of the nastiest diseases that we see in our clinic, namely the spectrum of fatty liver disease and dyslipidemia. And I like to think that translates into relative protection from atherosclerosis later on, but I can't make that case at the moment. Interestingly, however, other core components of the syndrome are intact and in fact more severe in these patients. So polycystic ovary syndrome, acanthosis nigricans, colonic polyposis, and diabetes are severe. And so I'm not going to make any more sophisticated comments about polycystic ovary syndrome other than to say all forms of congenital and acquired insulin resistance that we see do give rise to severe hyperandrogenemia, ovulatory dysfunction with imaging appearances which are essentially indistinguishable from what is called common or garden polycystic ovary syndrome. The testosterone levels sometimes are spectacular. They can be entirely reversed by a high-dose GnRH agonist to turn off gonadotrophin, so it must be a synergy between the gonadotrophins and insulin. And interestingly, in infants who have no functional insulin receptor at all, and the most severe recessive form of insulin resistance, they get hugely enlarged ovaries with hyperandrogenemia. So I suspect that this is not driven even by the insulin receptor. So I like to think that hyperinsulinemia has adverse effects on the ovary, probably driven by the IGF-1 receptor. But again, this is based on these observations as you see them. So this is an area that we're very interested in understanding in more detail, and specifically in discovering whether we can in some way exploit this difference between proximal insulin signaling and distal insulin signaling to try to do something to mitigate at least the fatty liver consequences of common obesity-related insulin resistance. But there's quite a few steps still to be taken before we can do that. So this rather long and woolly statement denotes the start of the final part of the talk. So finally, I will give you what I'm sure you've been waiting for, which is the more blow-by-blow account of insulin signaling. So just to recap, insulin binds to a transmembrane tyrosine kinase receptor, which induces trans-autophosphorylation of the receptor itself and phosphorylation of its 2 key substrates, IRS1 and IRS2. And in fact, there are also IRS3 and IRS4. Now that's critical because it's to these tyrosine phosphates that the regulatory subunit of PI3 kinase binds. This is type 1A PI3 kinase by definition. And these 3 catalytic subunits combine with a regulatory subunit. The regulatory subunit transfers them to the membrane where they have access to PIP2 or phosphatidylinositol 4,5 bisphosphate, which they hydroxylate at the 3' position to produce trisphosphate or PIP3. PIP3 shouldn't be there without any stimulation going on, and it's only evanescent. So it's that a PIP3 which is critical in binding and nucleating the downstream effector arms of the pathway through their plextrin homology domains, and in particular these 3 AKT2 enzymes, or these AKT enzymes, which actually do most of the stuff involved in the cell. And this has been studied in great detail for many years, both in cells and animals, by some of those scientists who I've discussed, who we'll be hearing from later in the week. So what can human genetics add to this story? Well, sometimes it's just a proof of the prior animal studies. And there's been a rather lovely trio of reports of the same homologous mutation affecting each of the 3 AKT isoforms. So it's been established by prior studies that AKT1 is predominantly involved in somatic growth. AKT2 is the most metabolic of the isoforms, while AKT3 is mostly in the brain. This mutation in the plextrin homology domain gives these molecules an opportunity to be partly uncoupled from upstream tyrosine kinase activation because they're able to stick a little bit to PIP2 in the plasma membrane and not PIP3. And the degree of activation that produces is really very tiny. So I'm showing here some cells from a patient with one of the AKT2 mutations. And you see, compared to putting insulin on the cells, the basal phosphorylation is tiny. But nevertheless, that's enough to produce catastrophic clinical syndromes. So the first discovered was by Les Bieseker's group at the NIH, and you can see this horrible deforming skin and skeletal muscle and skeletal overgrowth, which is Proteus syndrome. We described patients with very severe hypoglycemia, with all the characteristics of hyperinsulinemia, but without insulin, coupled to some hemihypertrophy in 3 patients with this disorder. They all had the AKT2 corresponding mutation. And you can see here their PEG tubes, which are keeping them alive overnight, without which otherwise they would fall into deep neuroglycopenia. Corresponding to the AKT3 mutation, the same mutation is found in mosaic form in these patients with horrible asymmetric brain overgrowth, often requiring hemispherectomy. So I think there are 1 or 2 aspects of novelty to be drawn from those syndromes, but actually predominantly they confirm what had gone before in model organisms. PI3 kinase is a bit more complex, and it's more complex partly because it's a dimer, and there are 3 different catalytic subunits, 2 nearly ubiquitous and 1 predominantly in hematopoietic lineages. And there are 2 different genes and 5 different isoforms of the regulatory subunit. I can't possibly summarize the vast PI3 kinase literature, but suffice it to say that these PI3 kinases are vital in a variety of major diseases. They are among the most commonly mutated and activated genes in cancer, and that's been known for more than 10 years now. They are critically involved in insulin-stimulated glucose uptake, as established by both animal and chemical inhibitor studies, and they play key developmental roles in B-cell maturation, angiogenesis, and a host of other factors, as well as being involved, it seems, in longevity, as you've heard already. Again, I'm not going to trawl through a lot of prior mice studies, so I'll just pick a few highlights for you. We know they're vital because if we delete either P110 alpha or beta, that's lethal in mice, and equally, deleting all 3 products of the PIK3R1 gene is also lethal. Interestingly, removing only 1 or more than 1, but not all, of the regulatory subunits produces increased insulin sensitivity, and the argument to explain that is that this reduces free excess subunit and enhances the coupling of the catalytic subunit with the enzyme. Luke Antley's lab observed 16 years ago now that loss of the long form, P85 alpha alone, severely impairs B-cell development to produce A-gamma-globulinemia and a lack of B cells. And Bart van Hazenbroek's lab showed that if you knock in half of a dead copy of the P110 alpha itself, that does the things that you might expect. It produces obesity and insulin resistance and diabetes. Although a couple of years ago, he and Lazaros Fukas came back and said, hang on, but actually, if you make those mice old, if you take them into their 2nd year, into middle age and beyond, actually they're protected from all those things and they live for longer. So even in that core aspect of P110 alpha, the core catalytic subunit of PI3 kinase, there are major questions. And I could go on. So we looked at cells from our patient with short syndrome, confidently expecting that we would find an obvious defect in insulin signaling. The mutation that we looked at cuts off the 2nd SH2 domain, the C-terminal SH2 domain, in all 3 of the products of the gene. And the SH2 domains are the domains which couple to phosphotyrosine, so that you might expect would lead to some degree of impairment of association with phosphotyrosine complexes. You can see in the skin cells of our patient, there's lots of the short version. And in fact, if we use an antibody which picks up all the isoforms, you can see the short isoforms as well as the short, long isoform. And first of all, everything went fine. When we stimulated our skin cells with insulin and we looked to see whether the P85 had been stuck to the phosphotyrosine complexes, we saw that that was the case in our control cells, but not in our patient cells. But after that, it all got a bit confusing because we couldn't see any difference in insulin-stimulated PIP3 levels, and this was done with a mass spec assay. And even more perplexingly, no matter how hard we looked at the stimulation of phospho-AKT downstream from insulin, we couldn't discern any difference in activation. And we did this many times in many ways. And you can see some representative blots here from our proband and from 2 controls. We're even more perplexed when we overexpressed the mutant in some 3T3L1 mouse adipocytes. And we did this only after they differentiated. And we expected that this would blunt insulin-stimulated glucose uptake. And yet, when we added doxycycline to the cells, as you can see here, and the key comparison is between here and here, there was no difference in insulin-stimulated glucose uptake. So really, we're very perplexed. We were facing patients who were severely insulin resistant, but some fairly commonly used assays which didn't seem to work out. Now I have to go quite far off-piste and out of my comfort zone into immunology. And I just want to tell you briefly about what other groups have been doing. So 3 years ago, it was shown that a mutation in a single patient that takes out the P85 alpha subunit, because it's a stop mutation early on, but leaves the other 2 intact, produces people, humans, with agammaglobulinemia and no B cells. Metabolic phenotype isn't reported, but may still have been there. Conversely, more recently, 3 different families were described with essentially cancer PIK3R1 mutations. In other words, these are mutations that delete some of this region in between the SH2 domains, which is known to be required for basal inhibition of the catalytic subunit. And these were germline mutations in at least one case transmitted from parent to child. And those patients also had immunodeficiency, broadly similar to what was seen in the deficiency of P85 alpha. And yet they had no other discernible phenotype. Conversely, our short syndrome patients, when we went back and looked, we knew already they didn't have any clinical history suggestive of immunosufficiency, and we confirmed that their immunoglobulin levels were entirely normal. They had normal responses to vaccination and normal lymphocyte counts, so there seems to be a true difference in these. So that's a lot of stuff about these more complex regulatory subunits. I'll finish by showing you a couple of human syndromes resulting from mutations in the catalytic subunits themselves. First is a series of conditions that we discovered with others only about 3 years ago now, and this is a very fast-moving area of work in our lab, a very fast-moving translational project. And it's become clear that somatic mosaic activating mutations in P110 alpha, and these are the same mutations that cause cancer, if they occur in mosaic form, produces this horrible spectrum of segmental overgrowth. And there's a whole world of different genetic terminology in here which has historically subclassified different groups. The first patient we met was this one, who, as you can see, has enormous legs, which were large, lifelong, but a rather emaciated, cachectic upper body. And that's because she has the unconstrained growth in her limbs, but not her upper body. You can see that the phenotype is dominated by where the mutation happens to occur. So even where it's not massive in degree, it can be absolutely horrible and disfiguring when it occurs in the face, for example. It seems to occur mostly in mesodermal lineages, so fat and muscle are most prominently affected. And interestingly, we've never seen one of the strong mutations in the hematopoietic cells. So that's what happens if you activate P110 alpha. Colleagues in Cambridge and then later at the NIH also found activating mutations in P1 delta. And essentially, these mutations are the same types of mutations. They activate the lipid kinase activity in the same sort of way. And these are tolerated in the germline again, so there are multiple affected members of each family who have recurrent sinopulmonary infections, really quite nasty, life-limiting, suppurating lung infections. And their phenotype involves decreased B cells and T cells, low antibody titers, and impaired class switching. And that's not altogether surprising, because this is the catalytic subunit that is known to exist in the lymphoid, in the hematopoietic lineage. So that's where we are. I have to say, predominantly in the cancer and in the metabolic fields, I think I'm fair to say that there has been a general assumption that the coupling of P110 alpha and P85 alpha is the main form of the enzyme, the one that we should really be concentrating on. That's partly by default, however. And yet, our observation in humans now shows that actually activating P85 alpha in humans, even in the germline, looks very like activating P110 delta, and yet there's none of that dramatic overgrowth which occurs when you activate P110 alpha. Now, it may be that there's other explanations, but I find it difficult not to conclude that this is in vivo human evidence of some specificity in coupling, which has been difficult to observe in complexes. It's also clear that P85 alpha itself plays a non-redundant role in B cell development. and antibody class switching. And although our short-associated C-terminal SH2 mutations, which clearly cause severe insulin resistance in humans, whatever that perturbation is, it's not required for this function in B cells. And rather interestingly, although we don't have P110 beta activating mutations, there is some work in mice in which P110 beta has been knocked out in the liver, which implies that it may mediate effects on metabolism which don't require AKT signaling. So it may yet be that this is more relevant to interaction with P110 beta. And so I think this is not overturning many years of animal study. It's absolutely complementary to it and is helping to steer and calibrate those studies. And we're now pursuing our findings in short syndrome in a mouse model, which I can tell you, without showing the data, looks at first sight to be a very faithful model of what we see in the humans. So we know now that C-terminal SH2 domain mutations in PIK3R1 lead to modest growth impairment and severe insulin resistance, but no immunodeficiency. And that's quite different to activation of PIK3CD, that's P110 delta, or PIK3R1. Mosaic activation of P110 alpha, PIK3CA, and also P85 beta, PIK3R2, produces dramatic tissue overgrowth. And if it does affect the liver, it produces hypoglycemia as well. Interestingly, in 170 patients, we've never found any PIK3R1 mutations in overgrowth, again arguing that that is not the consequence of that mutation in vivo, although they are common in cancer. And finally, selective germline loss of P85 does broadly mimic the consequences of activating P110 delta. So really, that's three examples of where I think human genetics has had something interesting to say, which hasn't just replicated what's gone on in model organisms, but which I think does begin to realize the vision that Sidney Brenner had in his Nobel Prize lecture 10 years ago, when he said that actually these mouse studies are all very well, but essentially the future is skilled human phenotypers, in other words, doctors, coupled to very high throughput, powerful next generation means of looking at natural genetic variation. And I think this is the embodiment of those sorts of opportunities. So as ever, I'm merely the front of house man. I'm merely the spokesman for the people that actually have done all the work behind the scenes. We have a range of activities going on in the lab, which I haven't talked about today, of more or less translational natures, but I'll highlight Nuno, who is a wonderful postdoc, who's very techie in the best sense. He loves molecular tools and has been our key person in introducing new molecular tools into the lab. Eric and Chen have been working on the DNA damage side of things, Paul D1 in particular, while Isabel, Patsy, and Alex have worked on the short syndrome mutations, and Vicky and Rachel have been doing hugely efficient and impressive work on the segmental overgrowth project. So my very final slide. So one of the great joys of what I do is the opportunity to interact with so many people around the world. Both the excellent scientists who you can see listed there, but also the busy clinicians who actually, A, notice something a bit odd, and then don't try to press it into an algorithm or into a sort of bucket diagnosis, and instead get on the email and on the phone and ask questions, and that's a great joy. I've mentioned the people in the lab who've done their work already, and that leads me really just to mention Steve O'Reilly and also my colleague David Savage. So Steve, as I said, I think is the doyen of the world of single-gene insulin resistance. And the true smartness in all of this work is that he worked out it was worthwhile doing that back in the early 1990s, before it became almost de rigueur in those studying any sort of disease. And he's pursued that with great vigor and imagination over the years. But I think my real debt lies not just in his scientific mentorship, but also in the environment which he's created for us all in Cambridge, which is a very permissive environment, and above all, in the fact that he's essentially allowed several of those who've grown up with him to bud off and take a little bit of his territory and run with it themselves. And that's quite an unusual and very generous form of mentorship, for which I shall be eternally grateful. Thank you.